evening, everyone. So today, my group and I will be presenting a research conducted by Chi Zhang and many others. This article is titled Antioxidant, Hepatoprotective and Antifungal Activities of Black Pepper, also known as Piper nigrum essential oil. So black pepper is known as the king of spices, and it has been used beyond food flavoring. Many research has shown that black pepper essential oil has components that possess strong antioxidant effects. In this study, it addresses a major issue, which is maize. It is susceptible to mold growth, also known as aspergillus flavus. This mold causes spoilage and also produces toxic metabolites. So plant essential oils are gaining attention as a natural antifungal for food preservation. There is many research that are currently surfacing to study the bioactive compounds in food for industrial application. So the objective of this research is to evaluate the antioxidant, antifungal, and hepatoprotective effects of black pepper essential oil, which could be significant for developing new natural food preservatives and hepatoprotective products. So next will be presented by Danya.
As for material points, the plant that we will be using is black pepper or Piper nigrum, which specifically picked from Hainan province in China. For the chemicals, we will be using butylated hydroxytoluene (BHT), gallic acid, gluten, cytokine, propidium iodide 5, rhodamine 123, and ascorbic acid, which is vitamin C. For the kits, it is a biochemical kit that consists of catalase, glutathione (GSH), total superoxide dismutase (TSOT), malondialdehyde (MDA), total bilirubin (TBIL), ALT, which is pyruvate transaminase, aspartate aminotransferase (AST), and alkaline phosphatase (AKP. For the strains that we will be using is Aspergillus flavus, which is a type of fungi. And lastly, for animal food, which is used for animal testing, we will be using 20 ml mice with average 2 grams of their weight. For the next part method, I will continue with Latifa.
For the method part, the first step is we extracted a black pepper essential oil with hydrodistillation by boiling pepper powder in water for 5 hours using a Clevenger device. The oil was stored in dark vials to preserve its properties. After that, we conduct a phytochemical analysis to analyze the black pepper essential oil's composition. And then we continue to conduct 3 tests. First test is the antioxidant assays. This test is conducted to analyze the total phenolics and flavonoids, which are the antioxidants, and then we also test its ability to block lipid peroxidation by using beta-carotene and TBA assays. Second test is liver protection study, in which we used mice. We induced liver damage in mice with CCl4, or known as carbon tetrachloride, which is a toxic chemical, and treated them with black pepper essential oil. So after 6 days, we measured the liver enzymes and antioxidant levels in blood and tissues. So for the third test is antifungal action. In this test, we tested black pepper essential oil on mice that are infected with Aspergillus flavus. We tracked how it disrupted the fungal cell membranes and mitochondria, proving its potential as a natural food preservative. And lastly, we do a statistical analysis in which we use the ANOVA using GraphPad, PRISM, or SPSS. Okay, that's all from method, and we move on to Hazim.
Thank you. So for the expected outcomes, there are 5 parameters that we need to be explored. So the first one is the total phenolic, flavonoid, and proanthocyanidin content of the essential oil. So within the references, we found out that the flavonoids is about 0.118%, phenolics is 23.756%, and protoanthocyanidin is 12.011%. Within the diagram itself, it's not quite accurate. It's because the percentage itself is actually quite low, so it's actually hard to show. So in the second one, which is the antioxidant activity assay itself, so the reference that we're going to be using is, again, as previously stated, is BHT and BC. So these are the standard of the antioxidant activities where a sample will be referencing to. So as there are multiple antioxidant assays that have been ran, I'll just simplify it to say that it is equal in terms of the antioxidant properties as compared to the other 2 references. So the third one, which is the effects of BPO on kidney histoarchitecture. So I would like to refer to figure C here. So the control is the liver of the mice before any addition, and the second image there is the addition of CCl4 or carbon tetrachloride, which results in a buildup of scar tissues. And this process is then important to show the effect of antioxidant towards the tissue sample, and the effect of the lipid peroxide to the tissue, which is tissue damage. So the third image shows the in certain concentration, the liver tissue actually will able to be recovered and be normal as with the control issue. And with this particular concentration is 400 milligrams per kilogram of BPO or black pepper essential oil. So for number 4 here is the effect of BPO on biochemical parameters, which are as follows, which is increase in antioxidant enzymes and decrease in lipid peroxide. So antioxidant enzyme actually help react with the reactive oxygen species such as peroxide, and as well as peroxide, which then this enzyme will then attack on this reactive oxygen species, and then convert it into non-toxic chemicals. So lipid peroxide, on the other hand, degrades cell membrane integrity as well as damage tissue. So we can see the decrease in the lipid peroxide. So number 5 is the activity. Number 5 is the activity against aspergillus flavus from fumigation. So looking at the figure, right? This figure show that that the 50 microliter per milliliter concentration of BPEO that has been sprayed on the maize seeds shows that they completely inhibit the fungal activities, including sporulation. So this then shows the capability that this compound can be used for fumigant for maize seed storage as low concentration as 20 microliter per microliter. So this final one is the mechanism of the antifungal action of the BPEO assay itself. So in these parameters, we find out the damage is caused by the release of the A flavors content, which is the nucleic acid. So this is because the BPEO is permeable enough to seep into the cell wall, which then degrades the cell integrity, releasing its content outside. So that's it from me. I'll pass it up to Zaira again.
Thank you, Hazib. So now I'll conclude our overall presentation. In a nutshell, we could say that black pepper essential oil could be a safe way to keep food safe by replacing chemical preservatives and also support liver health or in supplements or medicines. As proven in the research, BPEO helped to repair liver damage in mice, which is caused by toxin, and also it helped to inhibit fungal activity in maize. That's all from us. Thank you.